Okay, so good evening everyone. Welcome back to another class. So today is the ninth live session of the RNA Biology course session. So as you know, that in these live sessions what we do is that we basically discuss some previous year assignment questions. We not only discuss the questions, we try to discuss the concepts behind those questions. So it's easier for us to solve different type of similar type of other questions, right? So that's what we have been doing for the last 8 live sessions, and we'll be continuing that in this 9th live session. My name is Aishi and I'm a PMRF research scholar from the Department of Chemistry, IIT Bombay. So we try to make the sessions as interactive as possible. So if you have any doubts, you can write it in the chat box, or you can unmute yourself and let me know. You can raise your hand. If I know the answer, I will tell you right now. If I don't, then I will read about it and get back to you. Okay, so we'll start today's session. So the first question for today is, which of these are always expressed? Regulatory non-coding RNAs, housekeeping non-coding RNAs, meiotic RNAs, mitotic RNAs. Which of these are always expressed? I will wait for 3-4 minutes. You can try answering the question, and then we'll move on to discuss the question and the answer. Okay. So the answer will be housekeeping non-coding RNAs. Okay. So what are housekeeping non-coding RNAs? They are basically fundamental constitutively expressed RNA molecules essential for basic cellular functions like translation, including ribosomal RNA, transfer RNA, small nuclear RNA, and small nuclear RNA. Okay. So they are RNA molecules that do not. What are non-coding RNAs? So they are RNA molecules that do not encode proteins, right? So, unlike messenger RNAs. But what is the difference between housekeeping and regulatory ncRNAs? So, NC or non-coding RNAs are broadly classified into housekeeping and regulatory categories. So, housekeeping non-coding RNAs are essential for fundamental cellular processes and are found in most cell types. Examples can be rRNA that forms the structural and catalytic code of ribosomes, which are essential for protein synthesis. Can be tRNA, delivers amino acids to the ribosome during protein synthesis. Can be snRNA that plays a crucial role in RNA splicing, the process of removing non-coding regions from pre-mRNA. Can be snRNA, participates in the processing and modification of other RNAs, including rRNA. And there are regulatory ncRNAs that play a role in regulating gene expression and other cellular processes. They can be miRNA, small RNA molecules that regulate gene expression by binding to mRNA, leading to its degradation or translational repression. Can be lncRNAs, long non-coding RNAs that can function as scaffolds, interact with proteins or regulate gene expression. Can be circ RNAs, circular RNAs that can act as microRNA sponges or interact with proteins. So, housekeeping ncRNAs are essential for cell viability and are highly abundant in cells. Dysregulation of non-coding RNAs, including housekeeping non-coding RNAs, can contribute to various diseases, including cancer, right? You all know the difference between DNA and RNA, right? Let me zoom into the slide. Yeah. So we have to recall that RNA has two key chemical differences from DNA molecule. RNA uses a ribose sugar that contains a 2' OH and it also uses uracil base in place of thymine in DNA. Okay. So these minor chemical differences result in major structural differences between molecules. So while DNA is held in a rigid structure of the alpha helix, RNA is usually single-stranded and therefore has much more variation in the shapes that it can adopt. Like RNA is largely divided into two types, one that carries the code for making proteins or coding RNA, which is also called mRNA, and non-coding RNA. So the non-coding RNA can be subdivided into several different types depending either on the length of the RNA or on the function. Right? So size classification. Uh, Size classification begins with the short ncRNA, which are almost 20 to 30 nucleotide long, which include microRNAs and small interfering RNAs. The small RNAs, ncRNAs up to 200 nucleotides, which include transfer RNA, small nuclear RNA, and small nucleolar RNA. Sorry. And long ncRNAs, which are greater than 200 nucleotide, which includes ribosomal RNA, enhancer RNA, and long intergenic non-coding RNAs, among others. Sorry. Now, cells access the information stored in DNA by creating RNA through the process of transcription, which then directs the synthesis of proteins through the process of translation. So, the three main types of RNA directly involved in the protein synthesis are messenger RNA, ribosomal RNA, and transfer RNA. The mRNA carries the message from the DNA, which controls all the cellular activities in a cell. If a cell requires a certain protein to be synthesized, the gene for this product is turned on and the mRNA is synthesized through the process of transcription. mRNA then interacts with ribosomes and other cellular machinery to direct the synthesis of the protein it encodes during the process of translation. So mRNA is relatively unstable and short-lived in the cell, especially in prokaryotic cells, ensuring that proteins are only made when needed. Then we have microRNA. So the post-transcriptional expression levels of many genes can be controlled by RNA interference, in which miRNA-specific short RNA molecules pair with mRNA regions and target them for degradation. Right. So the process is aided by protein chaperones called argonauts. So this antisense-based process involves steps that first process the miRNA so that it can base pair with the region of its target mRNAs. So once the base pairing occurs, other proteins direct the mRNA to be destroyed by nucleases. Okay. So at the steady state, the vast majority of human cellular RNA consists of rRNA, 90% of total RNA for most cells. Although there is less tRNA by mass, their small size results in their molar level being higher than rRNA. Other abundant RNAs such as mRNA, snRNA and snRNAs are present in aggregate at levels that are about 1 to 2 orders of magnitude lower than rRNA and tRNA. So certain small RNAs such as miRNA and piRNAs can be present at very high levels. However, this appears to be cell type dependent. LNC RNAs are present at levels that are 2 orders of magnitude less than total mRNA. Although the estimated number of different types of human LNC RNAs may have a very restricted expression pattern and thus accumulate to higher levels within specific cell types. So, for example, sequencing of mammalian transcriptomes has revealed more than 100,000 different long non-coding RNA molecules that can be produced compared with the approximate 20,000 protein coding genes, and the diversity and the functions of the transcriptome within biological processes are currently a highly active area of research. So RNA polymerase enzymes are required to carry out the process of transcription and are found in cells ranging from bacteria to humans. So all RNA polymerases are multi-subunit assemblies. So with bacteria having 5 core subunits that have homologs in archaeal and eukaryotic RNA piece. In this section, we will learn more about these details. So we have talked about the process of transcription a lot of times, right? From transcription initiation, how it takes place in several steps, several stages. To start with, the RNA polymerase holoenzyme locates and binds to promoter DNA. At this stage, the RNA-P holoenzyme is the close conformation, initial specific binding to the promoter by sigma factors of the holoenzyme, sets in motion conformational changes in which the RNA polymerase molecular machine bends and wraps the DNA with mobile regions of RNA polymerase playing key roles. Now, next, RNA polymerase separates the two strands of DNA and exposes a portion of the template strand. At this point, the DNA and the holoenzyme are said to be in an open promoter complex, and the section of promoter DNA that is within is known as transcription bubble. So the RNA polymerase catalytic code within bacteria contains 5 major subunits: alpha 2, beta, beta prime, and omega. To position this catalytic code onto the correct promoter requires the association of a 6th subunit called the sigma factor. So within bacteria, there are multiple different sigma factors that associate with the catalytic code of RNA polymerase that help direct the catalytic code to the correct DNA locations where RNA polymerase can then help initiate transcription. For example, within E. coli, sigma 70 is the housekeeping sigma factor, okay, that is responsible for transcribing most genes in growing cells, and it keeps essential genes and pathways operating. Other sigma factors are activated during certain environmental situations, such as alpha sigma 38, which is activated during starvation or when the cells reach the stationary phase. So when the sigma subunit associates with the RNA polymerase catalytic code, RNA polymerase has then formed the holoenzyme. So when bound to DNA, the holoenzyme conformation of RNA polymerase can initiate transcription. So once the transcription bubble has formed and transcription initiates, the sigma subunits dissociate from the complex and the RNA polymerase catalytic subunit continues elongation on its own. Now in eukaryotic cells, three RNA polymerases share the transfer of transcription. The first step in gene expression, RNA polymerase is responsible for the synthesis of the majority of RNA transcripts, whereas RNA polymerase 3 produces short structured RNAs, tRNAs and SS5S are RNA. RNA polymerase II produces all mRNAs and most regulatory and untranslated RNAs. So the three eukaryotic RNA polymerases contain homologs to the 5 core subunits found in prokaryotic RNA polymerases. In addition, the eukaryotic polymerase 1, 2, and 3 have 5 additional subunits forming a catalytic core that contains 10 subunits. So the core has a characteristic crab claw shape which encloses a central cleft that harbors the DNA and has two channels, one for the substrate NTPs and the other for the RNA product. So two pinchers called the clamp and the jaw stabilize the DNA at the downstream end and allow opening and closing of the cleft. For transcription to occur, the enzyme has to maintain a transcription bubble with separated DNA strands, facilitate the addition of nucleotides, translocate along the template, stabilize the DNA RNA hybrid, and finally allow the DNA strands to reanneal. So this is achieved by a number of conserved elements in the active site, which include the fork loops, ruddered wall, triggered loop, and bridge helix. So here you can see a comparison of all the three RNA polymerases from eukaryotes. They all are homologs and share a similar core complex shown in the gray and the tan regions. And the stalk region is also related structure in all these polymerases. So the polymerase 1 and 3 enzymes have more subunits than polymerase 2. They have more limited arrays of transcripts that they create and tend to permanently incorporate transcription factors into their core structures. Polymerase 2, however, has a much more diverse array of targets and therefore has a wider array of transcription factors that needs to bind with. Therefore, they are not permanently incorporated, giving a smaller polymerase structure. In the next section, we have all discussed RNA transcription and how these general transcription factors that are critical for the recruitment of RNA polymerase. Two are typically referred to as a TF2 class of transcription factors, either identified by letter A, B, C, and so on. So the class 2 gene transcription is regulated at various levels while assembling on chromatin before and during transcription initiation, throughout elongation and mRNA processing and termination. Also, activators and repressors has been reported to regulate transcription, including a central multi-subunit complex called the mediator that helps with the recruitment of general transcription factors and the activation of RNA polymerase II. Okay, so we'll focus on formation of GTAs that make the core pre-initiation complex. Okay, we'll not go into too much detail, but transcription of RNA polymerase to dependent genes is triggered by the regulated assembly of the pre-initiation complex. This formation commences with the binding of the TF2D to the co-promoter, and it's a large megalodon-sized protein complex with around 20 subunits made up of 14 different polypeptides. The Tata binding, box binding protein, and the TBP associated factors. TF2D was shown to adopt an asymmetric horseshoe shape with 3 almost equal sized lobes, exhibiting a considerable degree of conformational flexibility that leads to distinct conformations. So, Tata binding protein is the TBP component of the TF2D, binds with a specific DNA sequence called the Tata box. This DNA sequence is found about 30 base pairs upstream of the transcription start site in many eukaryotic gene promoters. And if inhibitors bind with TBP, it will enable to interact with the DNA and start the formation of pre-initiation complex. In the absence of inhibitors, the TF2D will bind with the DNA and the Tata box binding protein will scan the DNA for the Tata box sequence. And when TBP binds to the Tata box within the DNA, it destroys the DNA by inserting amino acid side chains between base pairs, partially unwinding the helix and doubly kinking it. So note that TBP is released from TF2D during this process. So we will not go into too much detail. We know about the pre-initiation complex. We will talk about the natural elements which are basically important for the initiation of transcription. Promoters are defined as DNA elements that recruit transcription complexes for synthesis of coding and non-coding RNA. So enhancers are defined as DNA elements that positively regulate transcription at promoters over long distances in a position and orientation independent manner. So most of the time, enhancers will bring the RNA polymerase II complex to the target gene area to mediate transcription. The mediator complex shown in gray over here helps the looping process occur. However, sometimes enhancers may also lead to the short of transcription of non-coding RNAs from the enhancer region as well. So the function of the short non-coding RNAs is still not explored. So once transcription has been initiated, the elongation phase is where the RNA polymerase enzyme will use the template strand of the DNA to create the nascent RNA. And this diagram basically shows the catalytically active RNA polymerase. You can see that within the enzyme catalytic center, a small transcription bubble forms where the DNA helix is wound. So the template DNA strand is shown in blue over here, and the Mg2 plus cofactor is shown in red position at the catalytic active site. Okay. Sometimes the elongation phase does not move smoothly in the forward direction. The elongation complex can sometimes regress in a process called backtracing. So this can sometimes be caused by misincorporation or be caused the RNA polymerase complex. The backtracing process, however, stalls the elongation process, and it must now be rescued by additional protein helpers. So GRE factors are involved in rescuing a stalled transcriptional elongation complex. During normal elongation, the GRE protein is bound to the active elongation complex but does not exert any activity on the complex. It essentially stays out of the way like a tool in a tool belt. However, upon backtracing or nucleotide misincorporation, the GRE factor produces its own trigger loop domain, which supplants the normal catalytic trigger loop of the RNA polymerase enzyme that we saw in more detail in a previous slide. And the GRE factor will then cleave off any overhanging mRNA from the backtracked complex, and the elongation complex is then rescued, and then the normal trigger loop domain from the RNA polymerase will again be functional and can resume the elongation process. Termination of transcription in prokaryotes can occur using an intrinsic model. So the diagram here shows the polymerase in yellow and the open conformation with the DNA in blue and the nascent RNA in red. Intrinsic termination occurs at specific template sequences and inverted repeat followed by a run of A residues. And this sequence causes the formation of a short stem loop structure in the nascent RNA chain, which in essence will derail the polymerase from continuing. Extrinsic factors can also be involved in the termination process. This can mostly be aptly seen by the functioning of the RHO rho protein. As the nascent mRNA extends outward from the RNA complex machinery, the rho protein is a small horseshoe-shaped protein that can clamp onto the extending nucleic acid, and biochemical and structural data suggests that initially binds to the RNA in open lock washer conformation that closes interplanar ring as RNA transfers to the central cavity. There, the single-stranded RNA contacts an asymmetric secondary binding site. Upon hydrolysis of ATP, the RNA is pulled upon conformational changes of the conserved Q and R loops of the S base, leading to rho translocation and ultimately promoting rRNA polymerase dissociation. In eukaryotes, termination of protein coding gene transcription by RNA polymerase II usually requires a functional polyannihilation signal, typically a variation of the AAU-AAA hexamer. So nascent pre-mRNA is cleaved by the YSH1 protein and then polyannihilated by the CPA proteins. Currently, the details of this mechanism have not been fully elucidated and multiple models have been postulated. So, after prokaryotic mRNA processing, so bacterial cells do not have extensive post-translational modification of mRNA primarily because transcription and translation are coupled processes. So, they lack the physical barrier of a nucleus which allows the transcription and translation machineries to function at the same time, right? Enabling the concurrent translation of an mRNA while it is being transcribed. So within the system, the NUSG protein plays a critical role, which has two separate domains, and the functions of the two of them are known. And the NUSG N-terminal domain has the capacity to bind to RNA polymerase, whereas the C-terminal domain can combine with the NUSE component of ribosomes, and these two functions of the NUSG enable transcription to be coupled with translation. NUSG C-terminal domain can also bind to Rho to terminate transcription. In multicellular organisms, almost every cell contains the same genome, yet complex spatial and temporal diversity is observed in gene transcripts. This is achieved through multiple levels of processing leading from gene to protein, of which RNA processing is an essential stage. Following transcription of a gene by RNA polymerase to produce a primary mRNA transcript, further processing is required to produce a stable and functional mRNA, mature mRNA product. This involves various processing steps including RNA cleavage at specific sites, intron removal, cloth splicing, which substantially increases transcript repertoire and the addition of a 5' cap. Another crucial feature of the RNA processing of most genes is the generation of 3' ends with through initial nucleotic cleavage, followed in the most cases by the addition of a poly A tail, a process termed 3' and a cleavage and polyadenylation. Now, polyadenylation is required step for the correct termination of nearly all mRNA transcripts. In addition to determining the correct transcript length of the at transcription and its termination, the poly A tail fails to ensure the translocation of the nascent RNA molecule from the nucleus to cytoplasm, enhances translation efficiency, and acts as a signal feature for the RNA degradation. RNA cleavage and polyadenylation is carried out by a multi-subunit 3' end processing complex, which involves over 80 trans-acting proteins comprised of 4 core protein subcomplexes, and polyadenylation serial sequences in the nascent mRNA initiate the cleavage and polyadenylation process where approximately 50 to 100 adenosine residues are added to the mRNA. In eukaryotes, the 5' cap found on the 5' end of the mRNA molecule consists of a guanine nucleotide connected to the mRNA by an unusual 5' triphosphate linkage. And this guanosine is methylated on the 7th position directly after capping in vivo by methyl transferase. It is referred to as 7-methyl guanylate cap, abbreviated M7G, and there are some other unusual 5' caps, where the 7-methyl guanylate cap is the most common. For capping with 7-methyl guanylate, the capping enzyme complex binds to RNA polymerase 2 before transcription starts. As soon as the 5' end of the new transcript emerges from RNA polymerase 2, the CEC carries out the capping process. Enzymes for capping can only bind to RNA polymerase 2 with engaging in RNA transcription, ensuring specificity of the M7G cap almost entirely to the mRNA. So the 5' cap has 4 main functions: regulation of nuclear transport, prevention of degradation with nucleonucleases, promotion of translocation, C ribosomes and translation, promotion of a 5' proximal intron excision. In addition to the poly A tail, nuclear export of RNA is regulated by the cap binding complex, which binds to 7-methyl guanylate capped RNA. So the CBC or the cap binding complex then recognized by the nuclear pore complex in the mRNA exported. On cytoplasm, after the pioneer round of translation, the CBC is replaced by the translational factors EIF4E and ELF4G of EL4F complex, and this complex is then recognized by other translational initiation machinery, including ribosome, aiding in translation, including ribosome. Yeah. The 5' cap prevents degradation by functionally looking like the 3' end of the RNA and evading the 5' degradation next nucleases. Secondly, when the 3' end of the when the 5' cap is bound by translational machinery, it hides the cap from the decapping enzyme and increases the lifespan of the message. Now, decapping of a 7-methyl guanylate capped mRNA is catalyzed by the decapping complex made up of DCP1 and DCP2, which must compete with ELF4E to bind the cap, and the decapping enzymes are aided by enhancer proteins such as EDSC3 that can bind with mRNA in a sequence-specific manner and help recruit the decapping enzymes. So the 7-methyl guanylate cap is a marker of an actively translating mRNA and is used by cells to regulate mRNA half-lives in response to new stimuli. During the decay process, our mRNAs may be sent to P-bodies. P-bodies are granular foci within the cytoplasm that contain high levels of exonuclease activity. Eukaryotic organisms also have large tracts of non-coding regions interspersed throughout the gene sequences known as intron sequences. The introns must be removed from the pre-messenger mRNA before it translocates into cytoplasm to undergo the translation process. Right? So intron removal is mediated by a spliceosome, which is a macromolecular complex formed by 5 small nuclear ribonucleoproteins termed U1, U2, U3, U5, U6. We don't have to go into too much detail of all of this. Intron splicing, we have talked about all of these previous classes. We have talked about these also. So promoter is a specific short sequence of DNA at which RNA polymerase attaches and initiates translation at the beginning of transcription unit. So when RNA polymerase binds the promoter, translation occurs. Operator is a specific short of DNA sequence adjacent to the structural links that the repressor protein can bind to and prevent the transcription of structural genes. So they are encoded by repressor genes to regulate gene expression. So the activity of RNA polymerase is also regulated by interaction with ancillary proteins called activators, and the presence of activator removes repression and transcription occurs. So then the eukaryotic nucleus modified pre-mRNA before the genetic messages are dispatched to cytoplasm. As I said, at the 5' end of the pre-mRNA, a modified form of guanine is added, the 5' cap, which functions as protect mRNA from hydrolytic enzymes, and also is the translation start point for ribosomes. At 3' end, enzyme adds 15 to 15 RNA nucleotides, poly A tail. They act to protect mRNA from degradation by exonuclease, facilitate export of mRNA from the nucleus, terminate transcription. Information content of DNA in the form of specific sequences of nucleotides along the DNA strands. Proteins are the links between genotype and phenotype. So genes provide the instruction for specific proteins. And the bridge between DNA and RNA protein synthesis is RNA. DNA and RNA and protein synthesis is RNA. A specific sequence of hundreds or thousands of nucleotides in each gene carries the information for the primary structure of a protein. Linear order of 20 possible amino acids. So during RNA transcription, a DNA strand provides a template for the synthesis of a complementary RNA strand. Transcription of a gene produces messenger mRNA molecule. During RNA translation, the information contained in the order of nucleotides in mRNA is used to determine the amino acid sequence of a polypeptide. The basic mechanisms of transcription and translation are similar in eukaryotes and prokaryotes. Because bacteria lack nuclein, transcription and translation are coupled, which means as ribosomes attach to the leading end of an mRNA while mRNA transcription is still in process. In contrast, in eukaryotic cell, all translation occurs in the nucleus and translation occurs mainly at the ribosome in the cytoplasm. Okay. We'll not go into too much detail of the genetic code because these are other questions that we'll talk about it. We'll not go into too much detail of this also chat box. So which of these are already expressed? The answer will be housekeeping non-coding RNAs. Okay, that is the correct answer. I'll wait for 4-5 minutes for you guys. If you have any doubt, you can ask me right in the chat box. And then we'll move ahead with the next question. Okay. So next question. Okay. So dosage compensation do not cause balance in gene expression. Okay. So the options are true or false. Which one do you think is correct? I'll wait for a few minutes. And then we can move on to discuss the answer. So the answer will be false. Okay. So what is dosage compensation? But before that, we'll talk about the chromosomal theory of inheritance, right? So chromosome number is constant in all cells of a species but varies between species, right? Now, Saturn and ovary independently realize the parallel between Mendelian inheritance and chromosome transmission. and proposed the chromosome theory of inheritance, which states that Mendelian factors are located on chromosomes. Now, behavior of sex chromosomes offers support for the chromosomal theory. In many animals, sex chromosome composition relates to sex, but autosomes are constant. In both humans and fruit flies like Drosophila melanogaster, females have two X chromosomes, while males have X and Y. Males produce two kinds of gametes with respect to sex chromosomes and are called the heterogametic sex. Females produce gametes with only one kind of sex chromosome and are called the homogametic sex. So in some species, the situation is reversed with heterogametic females and homogametic males. So random fusion of gametes produces F1, that is half female, half male. See, Drosophila melanogaster organism is used extensively in genetic experiment. Okay. Inheritance pattern of X and Y chromosomes in organisms where the female is XX and the male is XY, and the production of the F1 generation. You can see female and male in the diploid parental genotype XXY, haploid gametes. F1 generation is, and then parent 1 gametes, F1 genotypes and F1 phenotypes. Inheritance pattern of X and Y chromosomes in organisms where the female is XX and the male is XY. Question of the F generation. Now, what is sex linkage? So Morgan found a mutant white-eyed male fly and used it in a series of experiments that showed a gene for Y color located on the X chromosome. First, he crossed the white-eyed male with a wild-type red-eyed female. All F1 flies have red eyes, therefore the white-eyed trait is recessive. Next, F1 were interbred. So they produced an F2 with 3470 red-eyed and 782 white-eyed flies. So the recessive number is too small to fit Mendelian ratios. Explanation discovered later is that white-eyed flies have lower variability. So all of the F2 white-eyed flies were male. Sex-linked inheritance of white eyes in Drosophila, red-eyed female and white-eyed male. So Morgan's hypothesis was thus that the eye color gene is located on the X chromosome. So if so, males are hemizygous because there is no homologous gene on the Y. The original mutant male's genotype was W and Y hemizygous with a recessive allele. Females may be homozygous or heterozygous. So the wild-type female in the original cross was W plus, and the F1 flies were W plus W and W plus Y. Females are heterozygous, males semizygous dominant. So the F2 data complete a crisscross inheritance pattern with transmission from the mutant fly through his daughter, who is heterozygous, to this transson. The F2 were this. So Morgan's hypothesis was confirmed by an experiment reciprocal to the original cross. A white-eyed female was crossed with a wild-type male, and without the reciprocal cross, all F1 females have red eyes, all F1 males have white eyes. So these F1 results are different from those in the original cross, where all the F1 had red eyes. So when the F1 from the reciprocal cross interbred, the F2 were this. So this is the reciprocal cross of that shown in figure 11.3, homozygous white-eyed female into red-eyed male. So Morgan's discovery of X-linked inheritance showed that when results of reciprocal crosses are different and ratios differ between progeny of different sexes, the gene involved is likely to be X-linked. So this was strong evidence that genes are located on chromosomes. So Morgan received the 1933 Nobel Prize for Physiology for this work. Now what is non-disjunction? So Morgan's work showed that crossing a white-eyed female with a red-eyed male produces an F1 of white-eyed males and red-eyed females. So his student found that about 1 in 2000 of the offspring was an exception, either a white-eyed female or red-eyed male. So this hypothesis was that chromatids fail to separate normally during anaphase of meiosis 1 or 2, resulting in non-disjunction. So non-disjunction can involve either autosomes or sex chromosomes, and for the eye-colored trait, X chromosome non-disjunction was the relevant event. So non-disjunction in an individual with a normal set of chromosome is called primary non-disjunction. So normal X chromosome segregation, non-disjunction in meiosis 1, non-disjunction in meiosis 2. So non-disjunction explains Bridges' findings. Non-disjunction rare event in a WW female would result in X with two X chromosomes and those with none. And if those were fertilized with normal sperm from a wild type male, the results were this. Okay. So the odd inheritance pattern matches specific aneuploid types, clearly associating a specific phenotype with the specific chromosome complement. So thus gene segregation mirrors chromosome behavior in meiosis, and Mendel's principles of segregation and independent assortment of genes correlate with the movement of chromosomes during meiosis. So parallel behavior between Mendelian genes and chromosome meiosis. So some mechanisms of sex determination include phenotypic sex determination in which sex is governed by genotype, environmental sex determination in which sex is governed by internal and external environmental conditions. So genotypic sex determination may occur two different ways. In the Y chromosome mechanism of sex determination, the Y chromosome determines sex, conferring maleness. In the X chromosome autosome balance system, the ratio between number of X chromosomes and number of sets of autosomes determines sex. So Y is required for the male fertility but does not determine sex. Sex of mammals is determined by the Y chromosome. In the absence of Y, gonads become ovaries. We're not going to do it now. What is the dosage compensation mechanism? Now, what is the evidence for Y chromosome mechanism of sex determination? So, understanding the Y chromosome mechanism of sex determination came from the study of individuals with unusual chromosome complements. In humans, these aneuploids include X of individuals who are sterile females exhibiting Turner syndrome. Now, most XO fetuses die before birth. Surviving Turner syndrome individuals become noticeable at puberty when secondary sexual characteristics fail to develop. Other traits include below average height, web-like necks, poorly developed breasts, immature internal sexual organs, reduced ability to interpret special relationships. So, XSY individuals are male and tend to be taller than average, fertility sometimes affected. X's individuals are usually normal women, although they may be slightly less fertile and a few have below average intelligence. Higher numbers of X and Y chromosomes are sometimes found, including XXXY, XXXXXY, and XXYY. Effects are similar to Klinefelter syndrome. Consequences of sex chromosome aneuploidy of humans are summarized in this table, if you can see over here. Now, what is the concept of dosage mechanism? The main question that we started with. Now, dosage compensation mechanism for X-linked genes in mammals. So, gene dosage varies between the sexes in mammals because females have two copies of X and males have one. So, early development gene expression from the X chromosome must be equalized to avoid death. Now, different dosage compensation systems have evolved in different organisms. In mammals, female somatic cell nuclei contain a barred body, highly condensed chromatin, while male nuclei do not. So, the lion hypothesis explains the phenomenon. Barred body is a condensed and mostly inactivated X chromosome. Lionization of one chromosome leaves one transcriptionally active X, equalizing gene dose between the sexes. An X is randomly chosen in each cell for the inactivation of early in development. In humans, 16-day post-fertilization. Descendants of that cell will have the same X inactivated, making female mammals genetic mosaics. Examples are calico cats, in which differing descendant cells produce patches of different color on the animal. Women heterozygous for an X-linked allele responsible for sweat glands, who have a mosaic of normal skin and patches lacking sweat glands. So, lionization allows extra sex chromosomes to be tolerated well. No such mechanism exists for autosomes, and so an extra autosome is usually lethal. The number of barred bodies is the number of X chromosomes minus 1. So X inactivation involves three steps: chromosome counting, determining the number of X's in the cell, selection of an X for inactivation, inactivation itself. Counting the chromosomes involves X inactivation center. Experiments in transgenic mice show that inactivation requires the presence of at least two XIC sequences, one on X chromosome. Autosomes with an XIC inserted are randomly inactivated, showing that XIC is sufficient for chromosome counting and initiation of hyalinization. Selection of an X for inactivation is made by the X controlling element in the XIC region. So there are different alleles of XCE, and each allele has a different probability that the X chromosome carrying will be inactivated. So the gene XIST is required for X inactivation. Uniquely, it is expressed from the inactive X, and the XIST gene transcript is 17 kb. Although it has no ORS, it receives splicing and a poly A tail. So during X inactivation, this RNA codes the chromosome to be inactivated and silences most of its genes. And inactivation itself is not well understood, but it's well known that it initiates at the XIC and moves in both directions, ultimately resulting in heterochromatic. So the gene of the Y chromosome determines maleness. So in placental animals, cells with the Y chromosome uniquely produce testis determining factor, which sets the switch of male to female development. The testis determining factor causes formation of testes instead of ovaries. All other sex differences result from the specific gonads, and so testis formation governs development of maleness. Studies of sex reversal individuals show that in XX males, a small fragment of Y is translocated to an X. Some XY females have a deletion of the same region of Y, and the human SRY, sex determining region Y, gene is in that region of the Y chromosome and has many of the expected properties. It is expressed only in the gonadal ridges of the embryo just before the testes form, and microinjection of the SRY gene into XX mouse cells produce normal males. The SRY gene product is likely a transcription factor regulating the expression of other genes involved in testis determination. Now, in case of Drosophila, an X chromosome autosomal balance system is used. Drosophila has three pairs of autosomes, the one pair of six chromosomes. Like humans, XX is female and XY is male. Unlike humans, Y does not determine sex. An XXY fly is female and the XY fly is male. The sex of the fly results in the ratio of the number of X chromosomes to the number of sets of autosomes. Okay. Now, in the normal female Drosophila, A equal to 2 and X equal to 2, the XA ratio is 1. In the normal diploid male Drosophila, A equal to 2, X equal to 1, XA ratio is 0.5. And in case of aneuploidy, abnormal chromosomal numbers, where the XA ratio is greater than 1, greater than equal to 1, the fly is female. The XA ratio is lesser than equal to 0.5, the fly is male. A ratio between 0.5 and 1 results in a sterile intersex fly with mixed male and female traits. Now, dosage compensation in Drosophila results in more expression of X-linked genes in males, so the level of transcription equals that of females to X chromosomes. So, a few species use environmental sex determination systems in which environmental factors affect the sex of progeny. Some types of turtles are an example. Eggs incubated above 32 degrees Celsius develop into females, while those below 28 degree develop into males, and eggs between these temperatures produce a mix of the two sexes. So details will vary with each species within the system. In the system, the environment triggers a developmental pathway which is under genetic control. So we will not go into much detail. So the theory of mammalian X chromosome inactivation proposed by Land in 1961 holds that almost all the genes on one of the two X chromosomes in the somatic cells of females are suppressed as a dosage compensation mechanism. The choice of either the paternal or maternal X chromosome for inactivation is random. So the inactivated X chromosome is identifiable as it is heterochromatic and initially has its DNA replicated later in mitosis than other chromosomes. The ZFY gene, thought to constitute the primary sex determining signal, was identified within a small segment of Y chromosome by Page and co-workers in 1987. However, the presence of similar gene ZFX on the X chromosome prompted the propounding of the dosage compensation inactivation theory of sex determination. So, according to this theory, both ZFX and ZF5 produce functionally interchangeable proteins, and it is therefore it follows that XY cells would have two active copies of the gene, while XX cells would only have one active copy due to X inactivation. Embryos with two copies would thus develop into males, while those with a single copy of the gene would develop into females. And in this way, the theory that held gene dosage determines sex. Though elegant, the theory became unattainable when it later became evident that ZFX gene escapes X inactivation, this contradicting the dosage compensation model. According to this theory, both ZFX and ZFY produce functionally interchangeable proteins. It therefore follows that XY cells would have two active copies of the gene, while XX cells would only have one active copy due to X inactivation. Embryos with two copies would thus develop into males, while those with a single copy of the gene would develop into females. In this way, the theory held that gene dosage determines sex. Right? So, though elegant, the theory became unattainable when it later became evident that the ZFX gene escapes X inactivation, thus contradicting the dosage compensation model. So, I've talked about dosage compensation. So, you can go through all these slides. I'm not going into too much detail. So, any mechanism in organisms with genotypic sex determination for equalizing expression of genes on the X chromosomes in males and females. So, organisms with X chromosomes have an inequality in gene dosage, the number of gene copies between the sexes. That is, there are two copies of the X-linked genes in females and one copy in males. In many such organisms, if gene expression on the X chromosome is non-equalized, the condition is lethal early in development. So normally, each gene is present in two copies. Departures from this condition, either up or down, can cause abnormal phenotypes and sometimes even death. So, how is the numerical difference of X-linked genes accommodated? Three mechanisms may compensate for this difference. Each X-linked gene would work twice as hard in males as it does in females. One copy of each X-linked gene could be inactivated in females. Each X-linked gene could work that half as hard in females as it does in males. And extensive research has shown that all the three mechanisms are utilized, the first in Drosophila, the second in mammals, and the third in the nematode Caenorhabditis elegans. So, in mammals, dosage compensation occurs by the decreasing transcriptional activity of X chromosomal genes in females to match that in males. And in Drosophila, the opposite occurs. So, transcriptional activity is increased twofold in males to match that in females, who have twice the number of X chromosomes. So, in both cases, chromatin remodeling is involved in regulating the transcriptional activity. So, the key male-specific lethal genes are MLE maleness, MSL1 male-specific lethal 1, MSL2, MSL3, and MOF. So, males with mutations in these genes die at the late larval stage, while females with the same mutations develop normally. The products of these genes are collectively called the male-specific lethal proteins. So the SXL late protein plays a key role in dosage compensation. In females, the SXL late protein binds to the transcript of MSL2, blocking its translation. The MSL2 protein is produced. In males, the MSL2 transcript can be translated because SXL late protein is absent. MSL2 forms a complex with the other MSL proteins, MLE, MSL1, MSL3, and MOF. And this MSL complex binds to about 35 chromatin entry sites on the Drosophila male X chromosome, and then MSL complexes spread from those sites in both directions into the flanking chromatin. The MOF protein of the MSL complex is a histone acetyltransferase, and its chromatin remodeling activity spreads along the X chromosome and is responsible for the twofold higher level of transcription of X chromosome genes in males than in females. So in females, the MSL proteins other than the MSL2 are produced. However, because MSL2 is essential for the binding of the MSL complex to the X chromosome, no chromatin remodeling can occur in X-sex females. So X inactivation, dosage compensation, and the expression of X-linked genes. So males 1 X chromosome, females 2 X chromosome. So here the mean amounts of the gene products of X-linked genes are the same in males as in females through X chromosome inactivation. So mechanism of X chromosome inactivation is XIC, X chromosome inactivation center, which controls expression of the XIST gene, X inactive specific transcript, which produces a non-coding 17 kb RNA molecule that codes the entire local X chromosome, cis-actin. Is located in XIC transcript only from the inactive X. The molecular mechanism behind X inactivation, the key player is the X-linked gene XIST. XIST is transcribed to produce a non-coding RNA that codes the X chromosome and inactivates it. It is uniquely expressed from the inactive X. XIST RNA does not travel over to any other X chromosome in the nucleus. Bar bodies are inactive X chromosomes painted with XIST RNA. So it's a key master regulator for X inactivation. It is expressed only from the allele on the inactive X. It is transcriptionally silent on the active X in both male and female cells. It has a function in the initiation phase of X inactivation. So transcription of XIST seizes on the other X chromosome, allowing all of its hundreds of other genes to be expressed. Shutdown of XIST locus on the active X chromosome is done by methylating XIST regulatory sequences. So methylation permanently blocks XIST expression and permits the continued expression of all the other X-linked genes. Okay, so I'll not go. So the compensation basically ensures an equal expression of genes from the sex chromosomes, even though females have 2 X chromosomes and males have only 1. So in each female, 1 X chromosome is inactivated and is highly condensed into a bar body, and females heterozygous for genes on the X chromosome are genetic mosaics. So British genetist Mary Leon discovered that in females, 1 X chromosome is randomly switched off, and this chromosome forms a dense body in the dense region in the nucleus called a bar body, and bar bodies are generally not found in males because the single X chromosome is still active. This explains why XXX females don't show symptoms. So the XIST gene on one of the 2 X chromosomes is randomly inactivated by DNA methylation, and the active XIST transcript and its RNA product codes the X chromosome. Histones on the coded X undergo methylation, which causes the chromosome to condense, forming a bar body and renders it inactive. So the coded uncoded X is left transcriptionally active. Inactivation of X chromosome occurs randomly in somatic cells during embryogenesis. Progeny of cells all have same inactivated X chromosome as original, creating mosaic individual. So inactivation is not always random. A structurally abnormal X is preferentially inactivated. Example: isochromosome X, extraembryonic membranes that go on to form the amnion, placenta, and umbilical cord in all the cells of the extraembryonic membranes. It is the father's X chromosome that is inactivated. Inactivation is not complete. Some genes are known to escape inactivation. That is those with a functional homologue on the Y. Example: genes located in the pseudoautosomal region. Still others are specific to X chromosome. Inactivation is not permanent, reversed in development of germ cells, not passed on to the gametes. So in normal female cells, the choice of which X is to be inactivated is random. Females are mosaic with respect to X-linked gene expression. Some cells express alleles on the paternally inherited X but not maternally inherited X. Other cells do opposite. So we'll not go into too much detail. We have discussed about dosage compensation and everything. I'll upload the slides so you can go through these slides that is based on the paper. So the answer will be dosage compensation do not cause balance in gene expression. That is false. It does cause balance in gene expression. So this question is done. If you have any queries regarding this question, I'll wait for a few minutes. You can type in the chat box and ask me, then we'll move on to the next question. So we'll move on to the next question for today. DNA in replication problem is alleviated by A. RNA polymerase, B. telomerase, C. reverse transcriptase, D. telomerase. I'll give you 2-3 minutes. If you can answer the question, then we'll move on to discussing this. So answer for this will be telomeres, right? So what are telomeres? Why are they important? Telomere shortening and end replication problem. All of this we'll discuss now. So these are telomeres. These are basically ends of linear chromosomes, right? And there is repetitive DNA sequences like TTA, GGG in vertebrates, and there are specialized proteins that form a capped end structure. Now telomeres cap the chromosome ends, right? So if you see, there is a telomeric loop that forms. So telomeres allow cells to distinguish chromosomes ends from broken DNA. Okay, like stop cells, repair or die. So the homologous recombination error free but need nearby homolog. Non-homologous end joining anytime but error prone. So why telomere is important? They prevent chromosome fusion by non-homologous end joining, right? So if you see that there is a fusion bridge, there is a breakage and fusion bridge breakage cycles, genomic instability, cell death or neoplastic transformation. So telomere also provides a means for counting cell division, like proliferative capacity and number of cell divisions, finite replicative lifespan and infinite replicative lifespan immortal. So how do cells know how many divisions they have completed? So there is an end replication problem. Telomere shortened with each S phase. DNA replication is bidirectional. Polymerases move 5' to 3', requires a labile primer, and each round of DNA replication leaves 50 to 200 base pairs DNA unreplicated at the 3' end. So telomere also provides a means for counting cell division. Telomere shortened with each cycle. Telomere shortened from 10 to 15 kb to 3 to 5 kb after 60 doublings. And cellular senescence is triggered when cells acquire one or a few critically short telomeres. So how do replicatively immortal cells avoid complete loss of telomeres? How do they solve the end replication problem? So this is an enzyme, telomerase, that's a key to replicative immortality. So it's a reverse transcriptase with RNA and protein components. It adds telomeric repeat DNA directly to 3' overhang, uses its own RNA as a template. The vertebrate repeat DNA on 3' end, TTA, GGG, and telomerase RNA template AAU, CCC. So telomerase, so how does it work? It overcomes telomere shortening and the end replication problem. It's expressed by germ cells, early embryonic cells. It's not expressed by most somatic cells, maybe expressed by some stem cells, but highly controlled. It's expressed by 80 to 90% of cancer cells. Remaining still need to overcome the end replication problem that do so by recombinational mechanisms. So, telomere length and cell division potential, if you see, the telomere length in humans and the number of doublets on the x-axis and the telomere length in humans on the y-axis. So, however, cells that express telomerase still undergo senescence in response to DNA damage, oncogenes, etc. So, what are the inducers of cellular senescence? Cell proliferation, short telomeres, DNA damage, oncogenes, strong mitogen, stress, potential cancer-causing events, etc. So what are the biomedical uses of telomerase? Expand cells for replacement therapies. Telomerase inhibitors to selectively kill cancer cells. And telomeres shorten with each cell division and therefore with age. Short telomeres cause cell senescence and senescent cells may contribute to aging. So telomere shortening causes aging and telomeres will prevent aging. That's a hypothesis. Now telomere length is not related to lifespan. Telomeres contribute to aging only if senescent cells contribute to aging. So telomerase protects against replicative senescence but not senescence induced by other causes. So telomeres are essential for chromosome stability. Telomere shortening occurs owing to the biochemistry of DNA replication, and short telomeres cause replicative senescence. Other senescence causes are telomere independent. So telomerase prevents telomere shortening and replicative senescence, and telomere hypothesis of aging depends on the cellular senescence hypothesis of aging. Okay. Now, initiation of DNA replication means primer synthesis and all of that. Different organisms use different mechanisms to make primers. Different fighters infect E. coli using quite different primer synthesis strategies. Coli fighters were convenient tools to probe DNA replication as they are so simple. They must rely primarily on host proteins to replicate their DNAs. So primosome refers to collection of proteins needed to make primers for given replicating DNA. So we'll not go into primer all of these. We'll directly go to telomerase. So at the ends of eukaryotic chromosomes are special structures called telomeres. Now one strand of telomeres is composed of tandem repeats of short G-rich regions whose sequence varies from one species to another. Now G-rich telomere strand is made by enzyme telomerase. Telomerase contains a short RNA serving as template for telomere synthesis, and C-rich telomere strand is synthesized by ordinary RNA prime DNA synthesis. So this process is like lagging strand DNA replication, and this mechanism ensures that chromosome ends can be rebuilt and do not suffer shortening with each round of replication. So all eukaryotes protect their telomeres from nucleases and double-stranded break repair enzymes. Ciliates have TEBP to bind and protect the 3' single-strand telomeric overhang. Budding yeast has CDC13P, which recruits STN1P and T10-1P. Mammals have fission yeast also have a protein similar to TEBP binding specifically to SS telomeric DNA. Okay, so we'll not go into too much detail. These you can just check the slides when I upload the slides. The step by step of semi-conservative mode of replication we have already discussed in one of the previous classes, so I'm not going into details. So we'll focus on telomeres, which are structures at the ends of linear eukaryotic chromosomes. It consists of a series of DNA molecules, sequences, and specialized proteins that bind to them. And telomerase adds a unique sequence of the ends of eukaryotic chromosomes discovered in tetrahydronucleus. They are needed at the ends of chromosomes for several reasons. They prevent the chromosome ends from being ligated together. Telomeres prevent the ends of chromosomes from being attacked by exonucleases, and they attach chromosomes to the any lamina, and telomeres begin pairing or synapses of homologous chromosomes during meiosis. And telomeres can compensate for incomplete replication of linear chromosome ends. Telomerase action. I'm not going into too much detail again. You can go through the slides when I upload this. You have the Matthew Messelson style experiment. So what is the end replication problem exactly? At the very end of a linear chromosome, however, there is no adjacent stretch of replicated DNA to provide this crucial 3' OH group. So when the primer at the end of the chromosome has been removed, it cannot be replaced by DNA nucleotides. It produces a gap at the end of the chromosome, suggesting that the chromosome should become progressively shorter with each round of replication. Chromosome shortening would mean that when an organism reproduced, it would pass on shorter chromosomes that it had inherited. Chromosomes would become shorter with each new generation and would eventually destabilize. So this situation has been termed the end replication problem. Chromosome shortening does in fact take place in many somatic cells, but in single cell organisms, germ cells, and early embryonic cells, chromosomes do not shorten and cell destruct. So how are the ends of the linear chromosomes replicated? So the requirement of an RNA primer to initiate all the new DNA synthesis creates a dilemma for the replication of the ends of linear chromosomes called the end replication problem. This difficulty is not observed during the duplication of the leading strand template. In that case, a single internal RNA primer can detect the initiation of a DNA strand that can be extended to the extreme 5' terminus of its template. In contrast, the requirement for multiple primers to complete lagging strand synthesis means that a complete copy of its template cannot be made. So even if the end at the last RNA primer for Okazaki fragment synthesis yields to the final base of the lagging strand template, once this RNA molecule is removed, there will remain a short region of unreplicated ssDNA at the end of the chromosome. Although this shortening would only occur on one of the two strands of the daughter molecule, after the next round of replication occurs, both strands of the daughter molecule would be shorter. This means that each round of DNA replication would result in shortening of one or two of the daughter DNA molecules. Obviously, this scenario would disrupt the complete propagation of the genetic material from generation to generation. Slowly but surely, genes at the end of the chromosomes would be lost. This is the telomere lagging strand primer problem. Okay. So, as the lagging strand replication machinery reaches the end of the chromosome, at some point, primase no longer has sufficient space to synthesize a new RNA primer, and this results in incomplete replication and a short ssDNA region at the 3' end of the lagging strand DNA product. So, when this DNA product is replicated in the next round, one of the two products will be shortened and will lack the region that was not fully copied in the previous round of replication. So one solution is to use a protein instead of an RNA as a primer for the last Okazaki fragment at each end of the chromosome. In this situation, the priming protein binds to the lagging strand template and uses an amino acid to provide an OH, typically tyrosine, that replaces the 3' OH normally provided by an RNA primer. So by priming the last lagging strand, the priming protein becomes covalently linked to the 5' end of the chromosome. Terminally attached replication proteins of this kind are found at the end of the linear chromosomes of certain species of bacteria and at the ends of the linear chromosomes of certain bacterial and animal viruses. So by binding to the DNA polymerase and to the 3' end of the template, a protein provides the priming hydroxyl group to initiate DNA synthesis. In the example shown, the protein primes all DNA synthesis as it's seen for many viruses. For longer DNA molecules, this method combines with conventional original function to replicate the chromosomes. So telomerase solves the end replication problem by extending the 3' end of the chromosome. So ends of the eukaryotic chromosomes are called telomeres. The sequence, for example, human telomeres consists of many head to tail repeats of the sequences, generally composed of head to tail repeats of TG-rich DNA. So although many of these repeats are double-stranded, the 3' end of each chromosome extends beyond the 5' end as single-stranded DNA. And this DNA structure acts as an oval origin of replication that compensates for the end replication problem. This origin does not interact with the same proteins as other eukaryotic origins, but it instead requires a specialized DNA polymerase called telomerase. And telomerase enzyme was discovered in 1985 by Elizabeth Blackburn and Carol Ryder. They share the 2009 Nobel Prize in Physiology or Medicine with Jack Szostak, who along with Blackburn determined how the unique structures of telomeres protected them from degradation. So telomerase is a remarkable enzyme that includes multiple protein subunits and an RNA component, ribonucleoprotein. Like all other DNA polymerases, telomerase acts to extend the 3' end of its DNA substrate, but telomerase does not need an exogenous DNA template to detect the addition of new DNTPs like DNA polymerase. Instead, the RNA component of telomerase serves as the template for adding the telomerase sequence to the 3' terminus of the end of the chromosome. Telomerase specifically elongates the 3' OH of telomerase single-stranded DNA sequences using its own RNA as a template. As a result of this unusual mechanism, this newly synthesized DNA is single-stranded. So this region of RNA can anneal to the single-stranded DNA at the 3' end of the telomere. Annealing occurs in such a way that a part of the RNA template remains single-stranded, creating a primer-template junction that can be acted on by telomerase. So one of the protein subunits of telomerase is a member of a class of DNA polymerases that use RNA templates called reverse transcriptases. Its subunit is called telomerase reverse transcriptase or TERT. Using the associated RNA template, TERT synthesizes DNA to the end of the TER template region, but cannot continue to copy the RNA beyond that point. At this point, the RNA template disengages from the DNA product. rearrange to the last one nucleotides of the telomere and then repeats this process. So this is accomplished by lagging strand DNA replication machinery by providing an extended 3' end. Telomerase provides additional template for the lagging strand replication machinery by synthesizing and extending RNA primers. Using the telomerase extended 3' end as a template, the cell can effectively increase the length of the 5' end of the chromosome as well. So, extension of the 3' end of the telomere by telomerase solves the end replication problem. Although telomere is only directly indexing the 3' end of the telomere, providing an additional template for lagging strand DNA synthesis, both ends of the chromosomes are extended. Telomerase extends 3' end of telomere. Additional 3' end DNA can act as template for new Okazaki fragment. Preparing Okazaki fragment, telomere extension still has 3' overhang. Okay, so that is so we have discussed the telomeres and the end replication problem. So we will not go into more details after this. You can upload the slides, you can just check out all these. These are taken from books and slides online, uploaded online. So coming back to the question that we started with, DNA end replication problem is alleviated by telomeres. Okay. So this will not be telomeres, it will be telomerase. Sorry, there's some kind of issue. Let me just correct it. This would be telomerase, right? The reverse transcriptase that we are talking about, not telomere. So let me correct this. Okay, so the right answer will be telomerase. Okay. So if anyone has any doubt with this question, we can discuss now. If you don't have, we can stop the session for today and we'll meet again next Monday. I will upload the video, YouTube video. You can check out the recording of the YouTube video and you can also check out the slides that I will be uploading. Right, so if you don't have any doubts, we can conclude the session for today and we'll meet again next Monday. Okay. Okay. Thank you so much. We'll conclude the session.